Hello, hello. Welcome to Following Science, the podcast where curiosity meets discovery. I'm Ritu Raparo, your guide on this journey through the fascinating world of science. Now, today's episode on CRISPR and gene editing. We'll dive into what CRISPR is, its discovery, how it works, and compare it with other gene editing techniques. Let's dive in. Let's start with understanding what CRISPR is. CRISPR are clusterated, regularly interspaced, short palindromic repeats, are segments of genetic code found in the DNA of bacteria. These segments are part of the bacterial immune system. They protect against viruses by recognizing and cutting viral DNA. The discovery of CRISPR's potential as a gene editing tool began in 1980s when scientists observed these repeating sequences, but didn't really understand their function. In 2012, Emmanuel Charpentier and Jennifer Doudna revealed how CRISPR, paired with an enzyme called Cas9, could be used to cut down DNA at specific sites. This discovery revolutionized genetic research. But how does CRISPR really work? Well, think about it like this. CRISPR and Cas9 function like molecular scissors. The system uses a guide RNA, which is called gRNA, to direct the Cas9 enzyme to a specific location in the DNA sequence. The gRNA then binds to the Cas9 enzyme and guides it to the target site, where Cas9 makes a precise cut in the DNA. This process allows scientists to add, remove, or alter genetic material with high precision. The simplicity and accuracy of this method make it a powerful tool for gene editing, and honestly really useful in scientific research because of how much power it holds. Now, CRISPR has been successful in various fields, and it's used in various fields. In medicine, for example, it's been employed to treat genetic disorders like sickle cell anemia and beta thalassemia by correcting defective genes in patients' cells. And in agriculture, for instance, CRISPR has been used to create crops with desirable traits, such as drought-resistant corn and non-browning mushrooms, enhancing food security and reducing waste at the same time. Now let's transition into the next segment and compare CRISPR to different editing tools. Before CRISPR, gene editing was possible with techniques like TALENs, or transcription activator like effector nucleases, and ZFNs, zinc finger nucleases. Now, TALENs worked by designing proteins to build specific DNA sequences and induce cuts. ZFNs use finger domains to achieve similar results. But now CRISPR is used, and it's preferred too because of its simplicity, efficiency, and cost effectiveness. Designing gRNAs is easier and less expensive than creating custom proteins for TALENs or ZFNs. More than that, CRISPR's precision reduces off-target effects, making it a safer and more reliable tool for gene editing. Now that we've covered the basics of CRISPR and its comparison with other gene editing techniques, let's move on to discuss the real-world applications and implications of this revolutionary technology. CRISPR has achieved several notable successes. For instance, in 2020, scientists used CRISPR to treat a patient with sickle cell anemia by editing her bone marrow cells to produce healthy red blood cells. That honestly makes me wonder about the potential that CRISPR has. If it can help treat a patient with sickle cell anemia, there's no doubt on what bounds it will be able to do other things in the future. Now, moving on to what's currently happening. Ongoing clinical trials are exploring CRISPR's potential in treating various diseases. For example, researchers are testing CRISPR-based therapies for genetic disorders such as cystic fibrosis and muscular dystrophy. Additionally, CRISPR is being investigated for its potential in cancer treatment, where it is used to enhance the ability of immune cells to target and destroy cancer cells. The future implications of CRISPR are just fascinating. Speaking of which, let's go and talk about some future implications. CRISPR holds the promise of eradicating genetic diseases. By correcting faulty genes at the source, conditions like cystic fibrosis, Huntington's disease, and even some sort of blindness could be potentially cured. This transformative power and potential that CRISPR holds offers hope to millions of people affected by genetic disorders. Now, while we all don't really like to talk about the bad stuff, sometimes it's necessary. Now, the power of editing genes raises significant ethical and ecological concerns. Altering the DNA/genes of wild populations, such as editing mosquitoes to combat malaria, could have unintended consequences on biodiversity and natural evolution. At the moment, the long-term effects of such interventions on ecosystems are still unknown, but that just warrants for cautious and responsible use of the technology. Now, let's transition into talking about some regulatory and policy considerations. Regulation is crucial in navigating the ethical and practical implications of gene editing, and different countries are approaching this regulation for CRISPR in different ways. Starting with the United States, the Food and Drug Administration, or the FDA, regulates gene editing therapies, ensuring that they meet the rigorous safety and efficacy standards before approval for clinical use. China, on the other hand, has more lenient regulations, allowing for faster research and application of the CRISPR technology. However, this approach does have a lot of ethical concerns, as seen in the case of CRISPR edited babies announced by the Chinese scientist He Zheng-Q in 2018. This controversial experiment led to widespread disapproval and calls for stricter regulations. The European Union, on the other hand, adopts a more cautious approach, emphasizing the precautionary principle and strict oversight. This ensures that genetic editing applications undergo through evaluation for potential risks and benefits before approval. Of course, the need for international cooperation and harmonization of regulations is critical. Global guidelines and ethical standards can help balance innovation with responsibility, ensuring that CRISPR technology is used safely and ethically. Now, of course, CRISPR and gene editing offer incredible possibilities, but they also come with a lot of ethical and regulatory challenges. It's important that we balance innovation with responsibility because that will be key as we continue to advance in this field. That's it for today's episodes, folk. Thank you for tuning into this episode of Following Science. If you liked what you heard, be sure to subscribe, rate, and leave a review on your favorite podcast platform. Your feedback helps me bring you even better content. Stay curious, keep exploring, and remember, the adventure of science is never ending. Until next time, keep following science.